All right, so welcome to week 2 of Introduction to Psychology Online. This is Neuroscience and Behavior. This unit takes up 2 weeks in the course, primarily because there's a lot of material that is interesting and somewhat complicated. And so I like to break it up so that you're not sitting there in front of your computer listening to me drone on and on for, an hour, hour and a half. So all behavior, all emotions that you experience, all thoughts that you have, your ability to ride a bike, your body's ability to digest food, all of those things, all of these behaviors are biologically controlled through what's called the nervous system. You've probably heard of this before. The nervous system is a biological network that controls basically everything that happens in your body. From the complex thought and planning, thinking about, okay, how am I going to do well in this online course? What do I have to do when? What do I have to do tomorrow? All the way down to your toenails growing. All of those things are controlled to some degree by the nervous system. Now, the nervous system is like a vast computer network. You can think of the nervous system as similar to the internet. The internet is a vast network of computers all communicating with each other. And the nervous system is much the same way. And similar to how each computer makes up one little node within the internet, neurons make up, the individual cells make up the nervous systems. So a neuron is a very, very simple structure. And one of the sort of themes from this lecture is how we can take these very, very simple processes and use them to do very, very complex behaviors. So just a little bit of anatomy, sort of the basic structure of a neuron. A neuron has several parts. Here we see the cell body. And this is the life support center of the cell. If you think back to biology that you maybe took in high school, this is where the nucleus of the cell is. This is where the mitochondria is. This is where all the basic life support functions of the cell exist. Now, the function of a neuron is to transmit information from itself to another cell, to another neuron. We'll talk about what that information is in a minute. But to receive messages from other cells, the neuron has attached to its cell body what we call dendrites. Dendrites look like trees. Or fingers. They sort of reach out away from the cell body of this particular neuron, and they form connections with other neurons. The key component to dendrites is that they receive information. Okay. The cell then can send information, and it does this using a combination of the axon and the terminal branches. So the axon, you can think about this as sort of the network cable that leaves the cell body and carries information that it receives from other cells onto subsequent cells. You can think about it like an electrical cable. It sends messages away from the cell body, and it's covered in a myelin sheath. The way that you can think about this myelin sheath, it's sort of a fat. It's a layer of fat that sits over the top of the axon, and it functions to actually speed up communication. The message, which we'll talk about, is an electrical impulse. It's just a little tiny blip of electricity. And you can think about the myelin sheath similar to how you think about the rubber insulation around copper wire. The rubber insulation around copper wire helps to insulate it. It keeps that electrical charge moving along the copper wire, and the myelin sheath functions in much the same way. And again, we'll talk about this electrical charge here in a minute. Okay. At the end of the neuron are what we call the terminal branches. And this is where this neuron connects to other subsequent neurons. So if you imagine over here, there's another neuron, and its dendrites reach up and form a connection with the terminal branches of this neuron. Likewise, you can imagine over here, there are other terminal branches from other neurons forming connections with this neuron. Now, This message that gets sent, that gets passed between neurons, is what we call a neural impulse. We can also call it an action potential. It is a little teeny tiny electrical charge that is generated in the cell body, and then it goes all the way down the axon to the terminal branches, and it passes information to the next cell. Now, most of the time, and I say most of the time, when we're thinking about neurological time, we're talking about milliseconds. But most of the time, the neuron is in the off position. Neurons only have two positions. They can either be on, they can fire their action potential, or they can be off. This is sort of interesting if you know anything about computer code. All computer code is coded in zeros and ones, on and off, yes or no. And neurons function in a similar manner. So most of the time, the neuron is at rest. It's just sitting there, living as little neuron life. And it is negatively charged. So the inside of that cell body is negatively charged. Now, if you take physiological psychology, we'd spend a week discussing how this cell is negatively charged. But for the sake of argument, for the sake of simplicity, just imagine it's just negatively charged side of a battery. Okay. What happens is when it turns on, right? When it turns on, it fires its action potential, and this little blip of electrical activity travels down the axon to the terminal branches where it can communicate with the subsequent cells. Action potentials are these electrical impulses, and the cell itself, operates on this sort of all or none principle. It's either on, it fires its action potential, or it's off, it's not firing its action potential. Now, the actual process of generating that little bit of electrical current is what we call depolarization. Essentially, what happens is the neuron receives enough stimulation from its dendrites to cause it to fire its action potential. And the ionic nature of the cell changes. Essentially, positively charged ions flood into the cell, negatively charged ions flood out, and that change generates that little tiny blip of electrical activity, which then shoots down the axon to the terminal branches, where it passes on to the next cell. Here in the PowerPoint, if you're following along, I have a link to a video that shows a depiction of what this depolarization process looks like. So feel free, if you'd like, to just pause me for a second and take a look at that link. Now, I've said so far that the action potential runs down the axon, it moves down the axon, the electrical energy moves down the axon, and it passes that communication to the next cell. But I haven't really gotten specific as to how that actually happens. So neurons are what we call a chemical electrical organ. They're a chemical, they function through both chemical and electrical processes. We've talked about the electrical process. This is the action potential. This is the neural impulse. The chemical process is what passes that information from one cell to the next. So what you see here on the screen is a very, very close-up version of this little junction right here. This is one terminal button from this sending neuron, and this is the dendrite of the receiving neuron, right? So we have the terminal button here, we've got the dendrite here. Now what happens is when the action potential reaches the end of the terminal button, when it reaches the end of the end of the axon, the terminal buttons, it causes the release of what we call neurotransmitters. And we'll talk about neurotransmitters in a minute. Neurotransmitters are stored in these terminal buttons in what we call synaptic vesicles. You can think about these just like little bubbles of chemicals that insulate them from the outside world, and they just kind of hang out and they're stored there in the terminal button. What happens is when it receives enough stimulation, the action potential, the terminal button moves to the edge of the axon and then opens up and releases those neurotransmitters into what we call the synaptic cleft, which is the space between the axon terminal and the dendrite, the receiving neuron. Now, it's important to note that these two things don't actually touch each other. So what happens is these neurotransmitters, they're released into the synaptic cleft, they float over to the receiving neurons, dendrites, and then what they do is they plug into the dendrite using what's kind of like a lock and key sort of system. You may have heard someone at some point say something to the effect of, there are, specific receptors in the brain for nicotine, for example. There are receptors in the brain for serotonin or dopamine. Serotonin, dopamine, nicotine, these are all neurotransmitters. And these receptors are these sorts of little receiving spots on the dendrite of the receiving neuron. So again, this process from start to finish, the electrical signal, the sending neuron receives enough information from its dendrites, it generates an action potential, that goes down to the axon to the terminal button. The terminal button releases neurotransmitters into the synaptic cleft, and those neurotransmitters float across and bind to the dendrite of the receiving neuron. And then depending on how many neurotransmitters and which ones, this receiving neuron may or may not fire its action potential. And so this is the basic process of neuronal communication. Now, when we talk about neurotransmitters, these are chemical compounds that bind to these receptor sites on this sort of lock and key kind of mechanism, right? So essentially, you can think about them like little puzzle pieces. One neurotransmitter is one little puzzle piece, and on the other side, there's a place on that receiving neuron where that specific neurotransmitter fits. And if that neurotransmitter is present in the synaptic gap, it goes over there and it plugs itself into that receiving site, right? And there are generally two classes of neurotransmitters. They can have an excitatory function, right? So these neurotransmitters, when they bind to their receptor sites, they encourage the subsequent neuron to fire its action potential. And there are also inhibitory neurotransmitters. And these, when they bind, they encourage the subsequent neuron to not fire its action potential. A couple examples of neurotransmitters, examples like acetylcholine. Acetylcholine is involved in muscle movement. So you can think of, when you raise your hand up into the air, the act of raising your hand up into the air is a function of your brain sending electrical signals through your nervous system to the muscles in your arm in a coordinated way to cause them to contract and move in a way that will put your hand up in the air. And acetylcholine is involved in the communication of those motor neurons. We'll talk about that in a minute, of those muscles. Dopamine is a neurotransmitter that's involved with reward. It's involved with mood and sleep. So for example, anytime we have sort of a positive experience, our brain releases a little squirt of dopamine to various areas in our brain. I sort of use the example of my dog. When my dog sees me reach into the treat box, my dog's brain gets a little squirt of dopamine. Getting treats is a positive thing. We like that. And so our brain rewards us for that. Dopamine is involved in positive mood. But it's also involved in how some drugs function, which we'll talk about when we talk about states of consciousness. Interestingly, too much dopamine is associated with schizophrenia. There we'll talk again also about the symptoms of schizophrenia, but it's a psychological disorder that involves auditory and visual hallucinations as well as other kinds of symptoms. But it also, too little can lead to Parkinson's disease, sort of lack of muscle control. We've also heard of serotonin. Serotonin's involved in a lot of things like mood and sleep. Many people are sort of familiar with serotonin in terms of its relationship to depression. There's evidence that suggests that people with relatively low serotonin levels tend to be more likely to experience depression. So we say that serotonin is involved with depression. Now we can divide up the nervous system into two components. There's the central nervous system, which is the brain and the spinal cord. And then there's the peripheral nervous system. And the peripheral nervous system is everything else. This is all the nerves that connect your body to your brain via your spinal cord. Now, if we just look at the peripheral nervous system, we can break this up into two subcomponents, the somatic and the autonomic. The somatic is the voluntary, sort of consciously controlled aspect of the nervous system, right? This is the system that's involved when I ask you to raise your hand and you raise it, right? The somatic system is what is controlling those coordinated actions. You can think about things like speaking and walking and driving a stick shift. All of these things are to some degree controlled by the somatic nervous system. The autonomic nervous system, or the autonomic side of the nervous system, this controls everything else. These are all the things that are sort of relatively outside of your conscious control. Life support functions, the functions of your internal organs, these kinds of things, right? You don't consciously think about having your pancreas release insulin when there's too much blood, when there's too much sugar in your bloodstream, right? The autonomic nervous system controls that. You don't think about beating your heart, right? The autonomic nervous system controls all of that. And so we say that this is a dual system. Now, if we further break this down, right? So now we're breaking it down even further. The autonomic nervous system has two primary functions or states. There's the parasympathetic state and the sympathetic state. The parasympathetic state should be sort of the default state. We call this sort of the rest and digest state, right? This is normal, everyday, automatic functions of the body, right? This is, reducing heart rate, reducing blood pressure, conserving energy, putting energy into digesting food, putting energy into cellular repair, right? Oftentimes our cells get damaged. The DNA gets damaged, or various things happen in our body, and our bodies have to repair those damages. You can think about this in terms of healing, right? If you have a cut on your hand, when the parasympathetic nervous system is active, then your body is actively trying to heal that cut on your hand, right? Rest and digest. We can contrast this with the parasympathetic nervous system. The parasympathetic nervous system becomes activated when we are in conditions of stress or threat. The sympathetic nervous system functions to prepare our body for what we call fight or flight, right? So what happens is when you are in a state of sympathetic nervous system arousal, your heart rate goes up, your blood pressure goes up, your body's resources are diverted away from your immune system, away from digestion, away from those regular body maintenance functions, healing and the like, and they're diverted to the muscles. They're diverted to mobilize your body to be ready to deal with whatever extreme, or maybe not so extreme, stress you're experiencing, right? Many people have had this sort of experience before, right? If you've ever been in a car accident, if you've ever had to give a public, if you've ever had to do any public speaking, you might have experienced parasympathetic arousal. So you're sort of amped up and you have sort of increased heart rate and you're kind of tense and you're ready to go. So these are the two features, or the two states, functions of the peripheral nervous system. Now we talk about these as two discrete functions, but in reality, they're more like ends of a continuum, right? So if you are leaving the L and it's the middle of the night and some really, really creepy person on the L who just looks off starts following you, right? You're going to have a pretty high degree of sympathetic arousal. Like you're freaked out, you're ready to go, you're ready to run or fight or whatever you have to do. And you can contrast that with, you have to give a presentation for class. You might be nervous, you might be stressed, but the level of sympathetic arousal isn't the same as if, your life is potentially endangered. Right? And the same thing is true for parasympathetic arousal. It sort of varies on a continuum of complete calm to complete, physiological arousal. And by the way, in this lecture, when I say arousal, I mean physiological arousal, not like romantic arousal, which we can talk about. They're kind of similar processes, but I'm just talking about physiological activity. So here's a fun little diagram just to help you guys visualize the relationship. So we have the nervous system on the whole. It could be broken up in the peripheral and the central. And then within the peripheral, we can break it up into autonomic. These are, the unconsciously controlled aspects of the nervous system, and the somatic, voluntarily controlled aspects. Further break up autonomic in terms of sympathetic and parasympathetic. Now, in our nervous system, we have a lot of different kinds of neurons. Again, if this were a biological basis of behavior class, we could probably spend a couple weeks talking about all the different kinds of neurons. For today, we're just going to stick to the three big classes of neurons. So these are sensory neurons, motor neurons, and interneurons. Sensory neurons, as the name would imply, they carry information from the body to the brain. Typically from the body through the spinal cord to the brain. There's examples of these. You can think of when you put your hand under hot water. The reason why you know that the water is hot is because there are individual neurons in your skin, which we'll talk about 2 weeks from today, that are sending information to your brain that your brain interprets as hot or cold if you put your hand in ice water. Motor neurons, they go the opposite direction. So they carry information, they carry commands from the brain to the various muscles and glands in the body. So for example, if you've got your hand under the hot water and the water starts, as you do in Chicago in the wintertime when water's really, really cold, so you've got to give it a minute to let it heat up. And you've got your hand under there and you're testing it. And at some point the water gets too hot. The sensory neurons are sending signals up your arm to your spinal cord, to your brain saying, this is hot, this is probably going to burn us. And then your brain sends commands out, we should probably move our hand out of the way, right? And those commands go down, contract the muscles in your arm, and in a coordinated fashion, move your arm out of the way, or hand out of the way. Now, the last class of neurons are what we call interneurons, and you can think of interneurons as kind of way stations. They're just neurons that connect neurons with other neurons, right? So they're not sensory per se, they're not motor per se, they just connect different neurons, right? So you can imagine it's not like there's one neuron in your hand that senses heat that travels all the way up your arm to your spine and then terminates in your brain. It has one giant long axon. That's not really how it works. The axon probably terminates in an interneuron, which then continues to send that information up a sequence of interneurons until it gets to your brain, right? So if all of that made your head spin, don't stress. All you really need to know is that sensory neurons carry information from the body to the brain. Motor neurons carry information from the brain to the body. And interneurons, they just connect neurons together. Now, here's an interesting example of how sensory neurons and motor neurons work. And this example is what we call a reflex arc. There are some instances, and our bodies are developed in such a way that there are some instances where it is advantageous for us to be able to respond as quickly as humanly possible. So for example, electric shock. When we experience an electric shock, the longer we are subject to that shock, the more damage potentially to our body that shock can do. So we have this reflex built in that allows us to deal with or react to this electric shock very quickly. And it illustrates very well the relationship between some kinds of sensory and motor neurons. So here we've got the sensory neuron in red, we've got the motor neuron in blue, and we've got an interneuron right here. So when, let's say you've got some toast that's stuck in the toaster, your toaster is 50 years old and it's kind of, a little bit sketchy, and you put the spoon in the toaster. Now, as you're putting the spoon in the toaster, your body is sending information up your sensory neurons, and it's going all the way up into your brain for processing. Okay. Now, let's say all of a sudden you hit a wire and you get an electric shock. And what that does is it causes a really, really extreme amount of sensory information, right? A ton of, not normal, but a ton of sensory information shoots up your arm to your spine. And what this reflex arc does is it has this little switch where when there's so much information, right, coming up to the spine, it bypasses the brain and sends a command directly to the muscles in the skin to yank your hand back to avoid further damage. Okay, so this reflex is an automated response, right? Is stimulus information coming in from the hand is enough that it bypasses the brain and generates a motor response on its own. Okay, and this is really useful, right? This is really useful because the quicker that we can respond, the less damage that we will ultimately suffer. Now, this isn't to say that you don't have a sense of that conscious control. Again, when we talk about neurons, and we're talking about time in terms of neurons, we're talking about milliseconds, millionths of a second, thousandths of a second. To us in our sort of experience, all of this happens at the same time, right? But if you slow that down to the time frame of a neuron, this reaction, sending this motor neuron or this motor command out via this interneuron, is much, much faster than the amount of time it would take for this information to travel all the way up to the brain, and then for the brain to send a command back down all the way to the motor neuron. Now, neural networks are what we use to describe how neurons are connected together to perform complex functions, right? I said one of the themes of this week's lecture is that we can take this simple on-off principle of how a neuron works, and we can build those together into very, very complex neural networks. An illustration of this is what I call a visual thesaurus, or what I call. I like to use the visual thesaurus to explain an illustration of how semantic concepts might be connected in our brains. So I'm going to open this up now, and hopefully this will work for the video. Here we go. So I'm just going to type in the word zoo. And when I type in the word zoo, and you can do this on your own. I'll bring this over here. Well, that wasn't a very good one. Let's type in the word zebra. Oh, well, zebra's not really good either. All of these should be on. Huh. Well, let's try school. Here we go. Okay, this works better. So I typed in the word school. Usually zoo works for me, but I'm not sure why it's not working now. Right, so you've got the word school in the middle. And you can imagine that this illustration represents a neural network, right? So school, the neuron of school, which again, I'm simplifying. There is not one neuron in your brain that represents school. It's a whole neural network in and of itself. But for visual purposes, right, the word school is connected to all of these different ideas, right? You could, school of fish, swimming, a shoal, fish, right? You can think of schoolhouse, they're a teacher, the educational system. All of these things are connected to the word school. And this kind of represents how neural networks are connected to each other in our brain. There is a pattern of probably hundreds of thousands of neurons in your brain that are all activated when you think about the word school. And those neurons, that pattern, is connected to, in some way, all of these other concepts, everything that you experience, everything that you have associated through your entire life with the word school. So maybe you love school, so it's associated with positive feelings. Maybe it makes you think about an experience that you had in school. Maybe it makes you think about all of the different schools that you've been to, right? Maybe it makes you nervous because it's a lot of work, right? Whatever it is, this idea of school is represented by a cluster of neurons, and those clusters of neurons are connected to all of the other concepts and experiences that we've had associated with the word school. And together, these clusters of neurons develop or represent neural networks. Okay? Just to give you an illustration of how dense our neurons are, we're talking about in the brain alone something on the order of 200 billion neurons with 300 trillion different connections, right? So if you look in this brain, all of this gray is just densely packed cell bodies of neurons, and all the white are individual microscopic axons, those axons that connect the cell bodies to each other, right? So this gives you a sense of how complex these neural networks are. Things that we take for granted that are a result of these complex processes, vision, right? For vision to occur, light enters our eye, and it is converted into a neural impulse, which we'll talk about next week. But literally from that point, when the light is converted into a neural impulse, there are billions of different cells that are all working together to take that light information and turn it into a representation of a visual scene that we experience. There's a tremendous amount of processing that makes that happen. And again, we'll talk at length about vision when we talk about sensation and perception. Another illustration that I have here is, how do you know where you are? How do you not get lost? How does your brain know where you are in space? This link will take you to a podcast that is the subject of one of the discussions for this week. And it's a story about a woman who literally does not have, she has a rare disorder where she literally doesn't have the ability to at times know where she is, even if she's sitting in her own living room. A neurologist kind of goes through and talks about how we think the brain works in terms of telling us where we are. And it's pretty fascinating, so give it a listen. So, how do we know all these things about our brains? It used to be that we could only know things about the brain by looking at people with brain damage, essentially poking people's brains to see what happens, electrically stimulating the brain. If I take a little electrode and I put it into your brain and I stimulate your brain and your arm shoots up in the air, then I know that the place in your brain that I'm stimulating is associated with that kind of muscle movement for your arm. We can also selectively destroy portions of the brain, lesioning. Typically, we don't do this to people, although as you'll see, there are instances where we have to do psychosurgery to remove a portion of someone's brain for a legitimate medical reason, and then we can observe the effects on their cognition and behavior. But stimulating, plugging a little electrode in and stimulating an area of the brain versus lesioning, destroying a cluster of neurons and seeing how that affects cognition and behavior. Current technology, currently we look at non-invasive methods to measure and study brain activity. So EEG, electroencephalogram, measures the electrical output of the brain at the scalp level. So, if you take the idea that the brain is an electrical chemical organ, that neurons firing generate small amounts of electricity, we can use the electroencephalogram to measure that amount of activity at the level of the scalp. This is useful. It'll tell us about brain activity, and it gives us really good real-time data, right? So this data is captured in real time. So if I show you a picture that makes you feel queasy, I will see where on your scalp there you have the most activity, and I might conclude, well, that area of the brain is associated with the experience of disgust or something like that. But the thing about EEG that's not so good is that it doesn't really give us very good spatial information. So it gives good temporal information. It tells us when things are happening with a fair degree of accuracy. But it doesn't give us spatial information. It doesn't tell us where that's happening specifically in your brain. We just sort of get a general, well, we're getting increased activity over here above your left ear, that kind of thing. MRI, on the other hand, uses, many people have had an MRI. It uses atomic magnetism. Essentially, an MRI is a giant magnet, and it rotates around your body or your head or whatever piece that we're interested in. And it causes the cells to change in such a way that we can render very, very detailed two-dimensional images of the brain. So, for example, if someone has a stroke, and we want to know, a stroke is a blood clot in the brain that causes damage, and we want to know where did that stroke occur, we can use an MRI to look specifically in what area of the brain that occurred. Now, the downside to MRI, for those of you who've ever had one, when you take an MRI, when you have an MRI done, you have to be perfectly still. You can't move at all. And one of the problems with MRIs is that they give us really good spatial information. We know exactly where things are happening in the brain and what these structures look like. But they don't give us any temporal information, right? This is just like taking a series of pictures. It doesn't tell us anything about when things are happening in your brain. Now, there's the last kind that I'll talk about, or maybe one more, is functional MRI, which is a version of MRI that combines these snapshots, these two-dimensional images, with temporal information that measures blood flow. Essentially, when you are, using your muscles or when you're using your brain, your cells are consuming sugar. They're consuming resources. They're consuming oxygen and that kind of thing. And so the cells that are consuming the most oxygen get the most blood, right? They get blood diverted to them. The cells that are consuming the most sugar get blood diverted to them. And so fMRI measures blood flow. And so this tells us about where in the brain particular things are occurring. So if we show you, a picture of something really gross that makes you feel kind of queasy, and we look at your brain, we can see where in the brain, what cells are responding to that stimuli. And they give us an indication as to what areas of the brain are responsible for what kinds of behaviors and what areas are working together when we're talking about different areas. The last one is positron emission tomography. This measures brain metabolism. So again, the rate at which cells in the body are metabolizing sugar. Again, it gives us really good spatial and temporal, so very good, when things are happening and where things are happening. But it's not so good because it does require, we inject you with radioactive material because that's what it ultimately measures. Positron emission is used sometimes for diagnosing cancer. It allows oncologists to sort of look at where tumors are in the body and how active they are and that kind of thing. So that's it for today. We'll come back probably next week. I'll have another post with the second half of this lecture. And hopefully after that, you'll have all the information that you need to contribute to the discussions, as well as complete the assignment for this week. So have a good week, and I will see you again soon.